Hi everyone, and welcome back to our online lectures for bioinformatics. Apologize for the slight delay in posting this particular lecture. I've been having internet issues of my own, and so hopefully you guys are not having such issues with getting access to internet. Um, and I hope you guys are coping quite well with the quarantine and the shelter in place order. But let's kind of return to um, how we actually find genes responsible for a given disease or trait. And remember that we kind of spoke about two specific strategies for identifying genes related to a particular disease or a particular phenotype or trait that we happen to be interested in. Um, A lot of our current efforts have been in analyzing whole exome sequencing, and so identifying those rare genetic variants or genetic changes that have a very large effect in a particular patient or individual. And so remember back to our activity with our father and mother and our proband that was suffering from osteopetrosis, the fairly rare bone disorder. But the other way of identifying genes related to a particular disorder is to use what's called a genome-wide association study. And this involves what's called genotyping, or basically sequencing individual base pairs, but just hundreds of thousands to millions of them across the genome. And so instead of sequencing, whole exons or whole genomes, we now basically shrink the genetic material that we're looking at by only sequencing those single base pairs that we know vary across individuals. And this helps us identify specific common variants that have relatively small effects, but collectively those small effects and common variants together can lead to some complex disorders. And so the way we actually measure these genotypes or detect these genotypes is by using an experimental technique called DNA microarrays, which basically allow us to genotype common variants across the genome. And we've obviously already talked about using next generation sequencing to obtain DNA sequence data. And so this is for whole exome sequencing, whole genome sequencing. And so we're now kind of switching focus from those rare variants that have a fairly large effect, as is the case in something like osteopetrosis or something, to more common diseases or more common phenotypes or traits that we happen to be interested in. And you may be thinking that hey, I think I've seen headlines in the news, or it kind of scroll across my feed when I'm looking for particular articles about scientists identify a number of genes related to academic success, or hundreds of genes linked to autism, for example, or new asthma genes are discovered. You might be asking yourself, well, I wonder how this is actually done experimentally. And the answer to this is by using that genome-wide association study. That's usually how they're able to identify these hundreds to potentially thousands of genes that contribute to any particular phenotype of interest. And so all the traits that we may study or are interested in studying all kind of fall along this particular spectrum. And so in the case of rare disorders, there may be one specific mutation that could be causing that particular disorder. And if that's the case, then we would be inclined to use whole exome or whole genome sequencing, so harnessing the power of that next generation sequencing approach to find those extremely rare variants in that particular patient. But if we happen to be interested in a more common disorder, so say, for example, like asthma or obesity or type 2 diabetes or something like that, things that are more common in the general population, we would often take a genotyping first approach where we use these DNA microarrays to basically genotype hundreds of thousands to millions of single nucleotide variants across our genomes. And so we're all pretty familiar with single nucleotide variants, right? And so just to remind us, single nucleotide variants are sites in the DNA sequence where individuals differ at a single DNA base, right? So when we compare two individuals, for example, at the same position in their genomes, they would be carrying a different nucleotide. And so, for example, changing a T to a G allele at a particular position in the CFTR gene, has been shown to cause cystic fibrosis. And we are also familiar with the concept of single nucleotide polymorphisms, pronounced SNPs, right? And so this is just a SNV, a single nucleotide variant, that is at a particular frequency when we compare it at the population level. And so it's usually over 1%. And so over 1% is termed, not common, but it's not uncommon to see that single base pair change. These are the most common form of genetic variation. This occurs about 1 in every 1,000 DNA bases, but can vary between individuals. Now, most of these single nucleotide variants, have no apparent effect on any particular phenotype, but we do know that a few contribute to diseases and traits, and that's kind of what we're trying to determine with these genome-wide association studies. And so we know, because we've been studying, that there are several, different types of SNPs. We've been more focused on this particular portion because we've covered whole exome sequencing. And so we know that there are synonymous and non-synonymous changes that lead to changes in the both the expression of that particular gene, how it's expressed within the cell, but also, building that particular protein that has that function within the cell. And so genome-wide association study at a very basic level is sort of our attempt to correlate certain genetic markers with particular phenotypes. This is usually done in particular types of diseases, but is not necessarily constrained to that. This particular technique has successfully led to the identification of some important genes related to health. And as discussed in the previous slides, this more often identifies common variants that are associated with particular diseases and traits. And this still requires experimental validation. And so we need to, once we do identify, a SNP that is statistically associated with a given phenotype or disease, we need to actually functionally demonstrate, that is actually happening. And so with that, we kind of have a overall explanation of what a GWAS or a genome-wide association study is in a nutshell. In the next section, we're going to kind of explain how we actually do these studies and how we identify specific SNPs that are associated with particular phenotypes of interest.